Hello medicos, we are here to make your med life easier with our simplified notes. As you see that gram-positive cocci are Staphylococcus and Streptococcus. How do we differentiate between those two? Let's see them. Staphylococcus and Streptococcus. Both are gram-positive cocci. That means violet-colored spherical-shaped bacteria. But this arrangement is different. Staphylococcus are arranged in a clusters, whereas Streptococcus arranged in a chains. Both are differentiated mainly through the catalase test. Catalase test is nothing but a hydrogen peroxide. In presence of enzyme catalase, it liberates oxygen, which appears as bubbles. In a slide, if we see for Staphylococcus, it is positive and Streptococcus, it is a negative. So that is the catalase test which differentiates Staphylococcus from the Streptococcus. Let's see Staphylococcus. It is a very important essay question in our microbiology. Staphylococcus. First of all, classification. In classification, based upon the coagulase test, these are differentiated into the two types, coagulase positive, coagulase negative. Coagulase positive is Staph aureus, which is highly pathogenic bacteria, whereas coagulase negative Staphylococcus called as a cons. These are differentiated by the novobiocin susceptibility test. Novobiocin is nothing but an antibiotic which differentiates the cons. If it is novobiocin sensitive, the organism is Staphylococcus epidermis. If novobiocin resistant, the organism is Staphylococcus saprophyticus. Now let's see what are the virulence factors, what is the pathogenesis, and what is the lab diagnosis for the Staphylococcus. Virulence factors. These are the factors which cause the toxic to the persons. Mainly virulence factors, we have to remember three headings in any bacteria. First one is a cell wall adhesions, second one is a toxins, and third one is a enzymes. Cell wall adhesions are nothing but a peptidoglycan, teichoic acid, protein A. These are all cell wall antigens. Let's come to the toxins. Toxins are mainly four in a Staphylococcus. Cytolytic toxins. Cytolytic, that means are lysing the cell, mainly RBC cells. 4 are present: alpha, beta, gamma, and PV toxin, Pantone Valentine toxin. This toxin is the cells. This is the toxins. And next coming to the enterotoxins. These are 8 types from A to E and G to I. Enterotoxin is nothing but entero, affecting the GIT. That means it leads to the food poisoning. And next one, toxic shock syndrome toxin, TSST. Next one, epidermolytic toxin, Staphylococcus skin syndrome. That means it separates the upper layer from the lower layer of the skins, which is a sign, Nikolsky sign. Nikolsky sign. These are the toxins. And next one is enzymes. Enzymes, mainly you have to remember the coagulase enzyme. As I have already told you in classification, Staphylococcus aureus is a coagulase positive organism. Two coagulase are present, free coagulase and bound coagulase, which are helpful in a slide test. Okay, fibrinogen in presence of coagulates forms the clots. Next, pathogenesis. What is the pathogenesis of the Staphylococcus? First of all, colonization. The Staph aureus which entered into the body colonizes and adheres to the tissues and fights with the host defense mechanisms. That means a phagocytosis, opsonization. It survives in spite of the host-defined mechanisms and transport to the other parts, causing the pathogenesis, mainly cutaneous diseases. That means on the skin, cellulitis, abscess, multiple furuncles, folliculitis, and deep infection in any organs. Toxin mediated. I have already told you the toxins, Staphylococcus scaly syndrome, toxin shock syndrome, toxin. These are all the toxin mediated types. Food poisoning, it is a 2 to 6 hours incubation period. Food poisoning, as all if you know that of classification based upon the incubation period, it takes 2 to 6 hours, mainly through the milk and milk products. Predominantly vomiting is the sign. Next, the toxin stimulates the vagus nerve which activates the vomiting center, causing the vomitings. Toxic shock syndrome, it is a multi-system disease causing high fever, hypertension, vomiting, diarrhea, and rashes. Staphylococcus scaly skin syndrome, I have already told you exfoliate skin diseases. It separates a layer from the dermis, which is a Nikolsky sign positive for the Staphylococcus. These are all for the Staph aureus, but we have also seen the cons, coagulase negative Staphylococcus, that is Staphylococcus epidermis and Staphylococcus saprophyticus. Mainly you have to remember the clinical manifestation. Staphylococcus epidermis cause the prosthetic valve endocarditis. Staphylococcus saprophyticus cause the UTI diseases. Let's see the most important topic of the Staphylococcus, that is lab diagnosis. It can be asked as a short, but it is also an important thing in our essays. In any bacteria, you have to remember the specific headings, that is specimen collection, from which you are collecting the the sample. Specimens, pus, insuppurative lesions, sputum, blood, and in any case of meningitis, you can also collect the CSF. This is the specimen collection. And direct microscopy. Under the microscopy, as all of you know, it is a gram-positive bacteria. So it appears in a violet color and it is a cocci. So it is a spherical shaped. As I have also told you, these occur in a clusters. You have to write that one only. Violet color, spherical shaped, arranged in cluster bacteria. That is Staphylococcus aureus. Next, culture medias. Mainly on blood agar, it is a beta hemolytic. That means completely hemolyze the RBC. We have also seen the cytolytic toxins, alpha, beta, gamma. So these causes the blood agar beta hemolysis. And next, on nutrient agar, it appears as a golden yellow color. Selective media. You have to remember the selective media for every organism, which is very important in MCQs also. For this, you have to use a Ludlam's and also the mannitol salt agar. These two you have to remember. Ludlam's and mannitol salt agar. Biochemical reactions. Already you know two, catalase test positive, coagulase test positive. Mainly you have to write these two. And you can write the others, mannitol fermentation positive, gelatin liquefaction positive, tellurite reductions. Next, bacteriophage typing. Done for the epidemiological purpose. Source of Staph aureus infections. Lastly, you have to do the antibiotic susceptibility test, ASTs, determined by the Stokes method, which is helpful in knowing what type of treatment you have to give to the patient. Thank you. Like, share and subscribe.